Nice to see you everybody. Welcome back to our biological science class. Gene regulation, the main topic. Under gene regulation, in E. coli bacteria, till now we have discussed humans, animals, plants and etc. But today, in this video session, I want to explain to you the topic related of the bacterium. The bacterium name is E. coli, short form, full form Escherichia coli. In E. coli bacteria, this concept will be performed. This concept name is lac operon. Lac in the sense lactose. Operon nothing but operation, the process. Generally, E. coli bacteria to produce energy, especially for the transcription purpose, lactose molecule is must and should, or else glucose. Whenever the glucose is insufficient or unavailable, this lactose it should be undergoes for the transcription process. So that process schematically on the board represented. This is in the absence of inducer, in the sense off mode. This is in the presence of inducer, in the sense on mode. This is off mode, this is on mode. Then what are these? They have given on the board in the sense this entire structure together called structural gene and promoters combination, nothing but clusters of genes. This entire sequence is called clusters of genes. This is also clusters of genes. Then in the cluster of gene, what are there? In the sense P, it indicates promoter. I, it indicates repressor mRNA. Then P, promoter. O, in the sense operator. Then this Z, Y, A. These three are the main structural genes. These three are comes under structural genes. Z, in a sense, it releases beta galactosidase. It helps to split the lactose. Remember, Z releases beta galactosidase, which helps to split lactose molecule. The next one, Y. Y helps to release permease. This permease, it allows the lactose molecule into the cell of the E. coli bacterium, allowing purpose. Then A, from A, trans acetylase will be released. That will be finally helping or completing the process of transcription of lac mRNA. Then let's see first off mode or on mode. First off mode, let me see. In off mode, repressor mRNA, generally repressor mRNA, it should convert into lac mRNA. Then from lac mRNA, the transcription process should be completed. But here, whenever lactose is unavailable, lactose is absent, then this repressor, it will bind with operator. Whenever repressor binds with the operator, then the process of translation will be blocked, stopped. On promoter, mRNA polymerase will come and bind. This mRNA polymerase, it has to handle or control this entire process. But here, whenever repressor bind with operator, that RNA polymerase, it is unable to continue the process, perform the process. Then this situation is called off mode. In the absence of inducer, inducer is nothing but a lactose molecule. Lactose molecule act as inducer, don't be confused. Inducer nothing but lactose molecule. So this is off mode. Then coming to on mode, same slight change can be seen. This repressor mRNA and repressor is there. Whenever lactose is available in on mode, lactose is available, that lactose acts like an inducer. This inducer binds with repressor. Whenever inducer binds with repressor, then repressor becomes inactive. Here repressor is active. No one is there to bind with that. But here something is there, someone is there. That lactose molecule or inducer binds with repressor, then this repressor unable to bind with operator. Now operator is free, then this mRNA polymerase, it will complete the process automatically. Now this repressor mRNA is converted into lac mRNA. This lac mRNA finally undergoes for transcription, then the complete mechanism will be finished. So the small change, in off mode repressor is active or free, in on mode repressor is inactive, it is not free. Inducer, it is binding with repressor because it is not free, inactive. So this is the concept. These three are structural genes: operator, promoter. Here mRNA will be binding, RNA polymerase will be binding, and repressor mRNA will be blocked, not allowing further process. But here mRNA is free, then it will be allowing to the further process. I hope everybody understood. If you have any doubts, please send your doubts in the form of comments in the comment box. Thank you. Signing off. This is Mr. Karthikeyan.